It's always a pleasure to be at this meeting, to really get to meet the families and the caregivers. And, you guys are very inspiring to us. Okay, I'm going to see if I can make this work. There we go. So, my first question is, why do we study genetics? And you guys at this point are all experts on what Chiari is, but to that question, why are we studying it? Why are we interested? Well, first and foremost, it gives us a better understanding of kind of the underlying problem and kind of how did we get here. It also can potentially help us make diagnoses, particularly within a family, if we're able to find a gene and another family member is worried whether or not they might be at risk. If we know what gene is responsible, that can help us hopefully maybe get an earlier diagnosis. And then, at the end of the day, what's important for all of you is treatment and therapies and approaches to make you feel better, right? And if we can learn something about the biology, that also helps us think through potential therapies better. So there's lots of evidence in the literature over the course of many years that suggests to us that at least a portion of Chiari is genetic. So first and foremost, sometimes it runs in families. And you guys have heard that by some of the previous speakers. The Millerat paper was really kind of the seminal paper, which noted maybe about 12 to 13% of individuals with Chiari also have a relative who has similar symptoms. Twin studies also support the presence of a genetic basis, and in particular, looking at identical twins and even identical triplets and looking to see that they are more correlated than not. And then also, guilt by association. You've heard a lot about Ehlers-Danlos syndrome, Klippel-Feil, some of these other conditions where we already know those are genetic, and Chiari tends to travel with them. And then somehow, let me see what happened with my slides. There we go. A little bit out of order. So animal models, we heard about this yesterday as well with the Cavalier King Charles spaniels who have similar symptoms and similar issues. And so why do we think that tells us it's genetic? And that's because we share a lot of the same genes with animals, right? And then finally, let me go back. We've also, at least in here at Duke, before I was part of the team, they did a really in-depth analysis within families of some of the characteristics that we see. And they found, number one, even though we use it as part of the diagnostic criteria, the tonsillar position and herniation really is not heritable, but that posterior fossa volume actually is heritable. So now we think genetics is important, but admittedly, there are challenges to finding the genes. we've talked a lot about some of these co-occurring conditions. Sometimes people are symptomatic even if they don't have tonsillar herniation. So, or you can have tonsillar herniation and not be symptomatic. So all these things make it challenging. We also know age of onset probably is important as well. And this more recently has borne out to be the case. And that is, before puberty, we tend to see pretty equal numbers of boys and girls who are affected. But once we move into adulthood, we actually have a preponderance of women who are affected. So that sort of tells us, too, maybe there are different genetics and different mechanisms with pediatric onset versus adult onset. We also heard a lot about surgeries and what makes it successful. So, we know that surgical outcomes are also part of the heterogeneity, and that's probably telling us something about genetics. So we've heard a bit about this already, but just to sort of ground you for some of this stuff I'm going to keep talking about, there's a lot of ideas about how Chiari sort of comes to be. Two of the more kind of prevailing mechanisms are cranial constriction, which is where posterior fossa is small, and those we tend to call connective tissue disorder negative, and then cranial settling, which you have heard about at the conference as well, which tends to be more associated with connective tissue disorders. And so there's more and more evidence which is coming out to sort of support the presence or absence of these connective tissue disorders as part of an important underlying heterogeneity, particularly with respect to genetics. And you heard Dr. Henderson speak yesterday, and we know surgical outcomes are associated with that too. So, how do we find the genes? So at, its simplest description, we're looking for parts of our genome that are shared amongst people who have Chiari, but not among those who do not have it, right? And so one of the approaches we can take is called association analysis. So this graph I have here is what we call a Manhattan plot. So it's like the Manhattan skyline, and you look for the peaks, and where the peaks are, that's where we have the best evidence for a gene. So you can think about this association analysis really is just trying to help us get to a region. It's not really going to help us identify immediately what the variant is, genetic variant. So it's sort of like if you're trying to find somebody's house in Durham and you're driving down the freeway, there would be an exit sign for Durham, right? So that's kind of what this type of approach does, is it sort of says, okay, the house is in Durham, the house is in this region, but it's not telling you exactly where the house is, right? So it does kind of help us narrow the search space, but it's not real specific. So you've probably heard a lot about sequencing, right? And so sequencing is a much closer view. So now we're actually maybe within the gene of interest. So we're on the right street. So we're going to go house by house to see if we can figure out where you live, right? So that's kind of taking it from a big view to kind of a very close view, right? So it gives us a lot of precision, and the technology now is amazing, right? So we can sequence your entire genome now, and we're doing that a lot, or we might want to just look at a specific gene. So we can do that as well. So based on all these different types of approaches, what do we know about the genetics of Chiari? So our group, back in 2013, performed a GWAS study, so that Manhattan plot that I showed you. And what we found was when we stratified our families on the presence or absence of connective tissue disorders, that really helped us quite a bit. And you heard about the Beighton score. So we actually used the Beighton to tell us which families potentially had a connective tissue disorder, because not everybody has a formal diagnosis, right? But what we found was in families that did not have evidence for connective tissue disorders, that there were 2 genes in this one region that sort of popped up. So these are what's called growth and differentiation factor genes. And these genes we were really excited about because they have been previously associated with Klippel-Feil syndrome, right? And so Klippel-Feil is one of those usual suspects that sometimes Chiari travels with. So also in that same year, a Spanish group decided to just hone in on specific genes. So you heard earlier from Dr. Heiss about how the posterior fossa sort of develops over, time in the womb and so forth. And so, their hypothesis was genes that are involved in that developmental process probably are involved. And they also focused on connective tissue disorder negative families. So they did find evidence for several genes. Again, all made sense. They were all developmental. And then Dr. Heiss presented this paper earlier today, where their group looked and used sequencing in several Russian families. And these were big families with lots of affected individuals. And although they didn't find the exact gene, they honed in on particular regions. So these were some of the candidates that evolved from that paper. And so none of the studies really to date have specifically focused on families where connective tissue disorders are present. But, we do, as has been sort of described earlier today, there are some genes that are known for Ehlers-Danlos syndrome, right? And so a lot of those are some of the collagen genes. And so we'll talk about that here in a second. But before I move to that, I want to point out a fairly recent paper by Sadler et al., which really a lot of different groups sort of came together and pooled some of their families, and we did sequencing analysis. And what popped up from that study are these two kind of related genes, the CHD8 and CHD3 genes. And these genes are really important for kind of altering what our chromosomes sort of look like when we turn some genes on and off. But importantly, the patients who had variants in those genes were pediatric onset, and those children tend to have kind of larger head structures. So again, kind of pointing to a very sort of specific presentation clinically that was associated with these particular genes. So now moving to the collagen genes. So our group recently published this paper where we did targeted exome sequencing, and our idea was to cover those same developmental genes in that original or BZU paper, but also to look at some of the collagen genes. And so we did find evidence, unsurprisingly, for some of the collagen genes, and then also picked back up the FLT1 gene that had originally been identified. So what do these findings tell us? First of all, I hope you can appreciate, much like all the clinical presentations at the conference, this is complicated, right? So it's complicated clinically. There's also lots of genes that seem to be involved. And I think being able to sort of tease apart different subsets of patients that maybe look similar is really going to help us kind of find these genes. Many of the genes, again, unsurprisingly, are involved in bone and connective tissue growth. Whether or not you have connective tissue disorders seems to matter, and there are different genetic forms that appear to be associated with those. And also whether you're pediatric or adult onset. So where do we go from here moving forward? I think we still need to kind of examine carefully this connection between Chiari and connective tissue disorders. We really just need to come together, all the institutions, across the U.S. and internationally, to really pool all the samples we have and sequence more cases and families, both with pediatric and adult onset, because once we have a large number, this is where statistics, the numbers become important. And so for us to kind of overcome some of the clinical variability, I think we really need large numbers of cases and families. And then, one of the things we really haven't done much in the field of Chiari genetics is to actually show functionally, once we find these variants, well, what do they do? How do they work? And so there was a little bit of that with some zebrafish work in the Sadler paper, but I think moving forward, we need to do more of that. And so finally, I think if we can do genetic testing, we should be able to find, what surgeries and what therapeutic approaches might be best for different patients. So I'm going to stop there and just thank all my colleagues and collaborators. And again, thanks for letting me talk.